nitrates in soil represents a usable form of nitrogen for plants to grow. But what do we know about molecular nitrogen, you guys? Can plants use molecular nitrogen as a source of nitrogen to make amino acids, proteins, and nucleic acids? No, they can't. So this process of nitrate reduction all the way back to molecular nitrogen, we call this denitrification. We're losing usable nitrogen. Okay, so just put that on the back burner, you guys, because as I said, when we talk very briefly about the nitrogen cycle, we'll see that denitrification is part of the nitrogen cycle. Okay, and again, folks, just really quickly, do note that in the right-hand column is the more evolutionary advanced process of respiration called aerobic respiration, right? This is what we talked about last, pretty much all last lecture. So again, in aerobic respiration, the significant difference is that molecular oxygen acts as a terminal electron acceptor. We're going to make the maximum number of ATP, 38 ATP per glucose, right? And our end products will be carbon dioxide and water. Good. Okay, you guys. So then what we're going to do now is we're going to flip down to a quick discussion of photosynthesis. And the photosynthesis we've been describing when we're talking about cyanobacteria, algae, and green plants. That type of photosynthesis is called oxygenic photosynthesis because it generates molecular oxygen. A oxygenic photosynthesis requires the photosynthetic pigment chlorophyll A. And again, we consider that as a more advanced type of photosynthesis. It turns out that the first type of photosynthesis that evolved here on primitive Earth used a different type of chlorophyll called bacterial chlorophyll. And in the use of bacterial chlorophyll, water is not used as an electron donor. So consequently, you do not make molecular oxygen as an end product. And hence, the type of photosynthesis that we see when bacterial chlorophyll is used, since it does not generate oxygen, it doesn't make oxygen, this type of photosynthesis is called anoxygenic photosynthesis. So genic, to make. This means molecular oxygen, and an means without. So this process is not going to make molecular oxygen, so no O2 produced. And again, this was the first type of photosynthesis that evolved using a more primitive type of photosynthetic pigment called bacterial chlorophyll. And I'll just use one example. I'll use the same example that I have on your handout, you guys. So in this example, and again, it's not balanced. Again, we're going to be using inorganic carbon dioxide as a source of carbon to make our organic molecules. But again, with bacterial chlorophyll, we cannot use water as a source of electrons that will be donated to carbon dioxide. Instead, in this particular example, we're using hydrogen sulfide. And again, in the presence of light and the presence of bacterial chlorophyll, low energy electrons from hydrogen sulfide are going to be stripped, and in the presence of bacterial chlorophyll, light energy will be used to energize those electrons in this incredible process of photosynthesis. And eventually those energized electrons will be donated to carbon dioxide to reduce it to energy-rich organic molecules such as glucose. So that looks similar, right, to oxygenic photosynthesis. But remember, since our electron donor is not water, we won't make molecular oxygen. Instead, we're going to end up with, this is elemental sulfur. And indeed, some of the bacteria who still carry out this primitive process, these include the purple and green sulfur bacteria. As they're carrying out photosynthesis, they're accumulating sulfur in their cytoplasm, and you can actually see these little sulfur granules in their cytoplasm. Again, part of the end product of anoxygenic photosynthesis. The point here, folks, is that in anoxygenic photosynthesis, are we making molecular oxygen? No. Okay, so that's the point. So no O2 is made. All right? Now, in contrast, we just take a look at the right-hand column there. There's the more advanced form of photosynthesis, oxygenic photosynthesis. Oxygenic photosynthesis requires which photosynthetic pigment? Chlorophyll A, right? And the huge advantage, well, from our point of view, the huge advantage of oxygenic photosynthesis is now the cells can use, so oxygenic photosynthesis. What are we going to make in oxygenic photosynthesis, you guys, besides glucose? We're going to generate, we're going to make what? Oxygen. Yeah, oxygen is made. And the reason is, again, we start out with our inorganic carbon dioxide. But now with oxygenic photosynthesis, our photosynthetic pigment is more advanced, I guess we call it. It's chlorophyll A. And we use CHLA as our abbreviation. So in oxygenic photosynthesis, in the presence of light, so this is our energy source, and chlorophyll A, now the cells can rip off electrons from water. In the presence of chlorophyll A, light energy is used to energize those electrons, and again, through an incredible process, eventually the electrons are donated to carbon dioxide. So again, we get literally light energy being converted into chemical energy. But you guys, what happens when we strip off all the electrons, or we can think of it as hydrogen atoms from water? What do we make? So we're going to make oxygen, and hence oxygenic photosynthesis. Oxygenic photosynthesis. Okay. Now, the reason I wanted to make a table, you guys, is that on the lecture exam 2, traditionally this is an area where people get really confused. So let me try to give you some examples of how people get confused. The biggest one is in comparing anoxygenic photosynthesis and oxygenic photosynthesis. So let me give you a common mistake that people make. So if I ask you to describe significant differences between anoxygenic photosynthesis and oxygenic photosynthesis, this is often a common answer. So a student will write, anoxygenic photosynthesis does not require oxygen, and oxygenic photosynthesis requires oxygen. Is that true or false? It's false, right? Because require suggests oxygen is a reactant, right? But when we're talking about photosynthesis and we say anoxygenic, we mean molecular oxygen is not an end product, right? It's not produced. And in oxygenic photosynthesis, we're saying molecular oxygen is an end product. It's being produced. Do you see that, you guys? Okay. And then I totally understand why, because probably a few questions before, I've just asked folks to describe significant differences between anaerobic respiration and aerobic respiration. Now if the student says anaerobic respiration does not require oxygen and aerobic respiration does, is that a true statement? That's true, right? But I totally understand, you guys, how I would get them confused too, right? So remember, with respiration, we're asking, is O2 the terminal electron acceptor, right? When we're discussing photosynthesis, we're asking, is molecular oxygen an end product? Okay, so that's just that's a real common area of confusion. So hopefully we're okay with that. Okay. Righty then. Okay, so on your handout, you guys, you give some examples of organisms that carry out oxygenic photosynthesis. So can you give me 3 groups of organisms that make chlorophyll A and hence can carry out oxygenic photosynthesis? Cyanobacteria, good. Algae, good. And green plants. Okay, of those, which are eukaryotes? Algae and green plants, right? So of those three groups, who has chloroplasts that contain chlorophyll A? Good. That is right, good. Remember you guys, the cyanobacteria do not have chloroplasts, right? The cyanobacteria evolved into eukaryotic chloroplasts. I always have, I don't know why, I think I struggled with that, and I think it was because I thought, well, if you make chlorophyll A, you must have a chloroplast, right? But remember, bacteria, cyanobacteria, not only do they lack membrane-bound nuclei, they also lack membrane-bound organelles, such as mitochondria and chloroplasts. So a bacterium cannot have a chloroplast, right? And I had the toughest time just getting that under my belt. Okay. Now, folks, what we're going to do, the last topic in Chapter 5 that we're going to cover, we're actually stealing a little bit of information from later chapters. But what I wanted to do is just give one example of how important microbes are in the process of what we call an element cycling here on Earth. So the cycling of sulfur, the cycling of nitrogen, the cycling of carbon. So in your multi-page handout, the very back page has this very, very brief description of one example of element cycling that microbes play an important role in. And that is in nitrogen, the nitrogen cycle. Okay. So I'm just basically on the board going to put parts of this diagram. And do remember, folks, that on lecture exam 2, the only parts of the nitrogen cycle you'll be responsible for will be those that we're going to discuss today. Okay, so in your textbook and in the PowerPoint slides, let me just advance to there, you guys, so we'll blast through the photosynthesis slides. And I do want to apologize. Photosynthesis is such an incredible process. It's crazy, but sadly, because of time, we're not going to be able to go through all the steps. Okay, so it is a really fantastic process. Oh, and as we go, I can just maybe show you some tables from your book that might help you in preparing for lecture exam 2. So this, I thought, was a nice study aid, table 5.5 in your textbook. It's comparing aerobic respiration, anaerobic respiration, and fermentation. And you can just imagine how many wonderful questions I could pull from that, you know, comparing and contrasting these three processes. So that's a nice study guide. This was just to remind me that our discussion is probably really misleading. All we've done is focus on glucose as a source of carbon and energy. Those of you that have had A&P, those of you that have had nutrition, you know that cells can use more than just glucose, right? So this was just to remind me, to let you all know, we've barely scratched the surface with regard to metabolism. This is to remind me, to let you all know that, that many microbes can use proteins as a source of carbon and energy. They can use other sugars other than glucose. They can use lipids as a source of carbon and energy, right? So I just to let you know, you guys, that we have barely scratched the surface of metabolism. And this was just our slides to remind us of this more primitive anoxygenic photosynthesis. And it's kind of cool, you guys, here. This is one of the purple sulfur bacteria. And see these little yellow refractile? That's the sulfur granules that are accumulating as a result of anoxygenic photosynthesis. I thought that was kind of cool. And then, of course, our oxygenic photosynthesis. So, an example of our cyanobacteria. And this again, you guys, is just, we don't have time to go through the events of photosynthesis, but it is amazing. It's better than a movie. I mean, it's incredible. If somebody wrote this up and gave it to the Hollywood folks, the filmmakers would go, no, this is impossible. Forget it. You know, we're not going to make a movie about photosynthesis. And here we have a table again, helping to compare different types of photosynthesis. So we described that in algae and plants that make chlorophyll A, they're carrying out oxygenic photosynthesis. And you guys, in our discussion in cyanobacteria, I only emphasize oxygenic photosynthesis. It turns out that some can carry out anoxygenic, but on the lecture exam, I will only focus on cyanobacteria carrying out oxygenic photosynthesis. And here we see amongst our bacteria, we have some groups that carry out anoxygenic photosynthesis. So just again, another possible study aid. And then this is the topic we're going to finish with. In your book, it has, and in the PowerPoint file, there's many slides on the nitrogen cycle, but we are going to totally condense it into basically this one picture, okay? All right, so this is where we'll be ending. And again, the point here is we want to just briefly touch upon the important role of microbes in element cycling here on Earth. And because California agriculture is really important here in California, I thought we would focus on the nitrogen cycle and the role of microbes in the nitrogen cycle. And we want to take a look at how the nitrogen cycle can influence plant growth here on Earth. So we're going to start out with molecular nitrogen in the atmosphere. So this is N2. And in atmospheric air, molecular nitrogen is a major component. Oxygen, molecular oxygen, only represents about 21% of the atmospheric air. But, excuse me, help me out, folks. Can organisms use molecular nitrogen as a source of nitrogen to make amino acids, proteins, and nucleic acids? No. And the reason is this triple covalent bond is so hard to break. So the only organisms that can make molecular nitrogen usable are those that have an incredible enzyme complex called what? Nitrogenase, exactly. And nitrogenase reduces molecular nitrogen to ammonia. Now, ammonia is a usable form of nitrogen. Organisms can use ammonia to make amino acids, proteins, and nucleic acids. But again, we want to remember, very few organisms have nitrogenase. It's believed it evolved when Earth's atmosphere was still anaerobic. And the reason biologists think this is it is inactivated. It is inactivated by molecular oxygen. Some people say if something is not active in the presence of oxygen, they say it's oxygen sensitive, right? So you all know in lab we discussed how the cyanobacteria create an anaerobic environment by forming a special cell, the heterocyte, that will protect the nitrogenase from molecular oxygen made by the photosynthetic cells. And we discussed how the soil bacterium Rhizobium will invade the roots of legumes and trigger the legume to form these beautiful root nodules, creating an anaerobic environment in which the Rhizobium's nitrogenase can carry out nitrogen fixation. So this process, this first step, you guys, this is called nitrogen fixation. And as farmers, we love this. As farmers or gardeners, this is dynamite, right? Okay. Now, what we'll do, let's pretend this is air and this is soil or dirt. Now, it turns out that ammonia can be further processed under aerobic conditions in soil. If soil is nice and fluffy and airy and not saturated with water, then there are other soil bacteria that can further process ammonia to nitrite, and then what we really want for our plants are nitrates. My understanding is plants love nitrates as a wonderful source of usable nitrogen to make proteins and nucleic acids. So this process is carried out by soil bacteria. Is called nitrification. Okay. And this two-step process, we can see in this two-step process, we're taking unusable molecular nitrogen and converting it to this wonderful form that plants love. So if we have nitrogen fixation and nitrification occurring in our soil, let's say we're farmers, we have happy plants and we are happy farmers, right? Our crops are growing really fast, we're going to have really high yield. So we've seen how microbes play a role in both these processes. We have our Rhizobium, we have our cyanobacteria involved in nitrogen fixation, and soil bacteria involved in nitrification. But there is a dark side to the nitrogen cycle, and that is a third part of the cycle that occurs when we get saturation of our soil with water. folks, that in water, oxygen diffuses poorly. So if we have flooding, or if we have over-irrigation, meaning we water our fields too much, this will create an anaerobic environment. So we have an anaerobic condition in our soil, and we don't want that. One of the reasons is that it will trigger other soil bacteria to carry out processes such as anaerobic respiration. And you guys will recall in anaerobic respiration, what can act as an alternate terminal electron acceptor? Nitrates, right? And do you recall when we were talking about anaerobic respiration where nitrates is used as the terminal electron acceptor? We said the nitrate can be reduced all the way back to what? All the way back to molecular nitrogen. Now, can plants use that? No, they can't. So this process is referred to as denitrification. Denitrification because it represents a loss of usable nitrogen, right? So if we're farmers, we don't want this to occur because it's going to make our plants very sad. We're going to have sad plants, right? So we don't want denitrification occurring, right? So, and especially in a drought year like this year, you guys, this is just a reminder, we don't want to water our gardens too much. If we're farmers, we don't want to over-irrigate our fields because we'll create these anaerobic environments, trigger soil bacteria to switch from aerobic metabolism over to anaerobic respiration, and we're going to get a loss of usable nitrogen. Does that make sense? Okay. And it just occurred to me, folks, I forgot to mention one last thing with regard to aerobic respiration, anaerobic respiration, and fermentation. Some microbes can carry out all three processes, and a great example is E. coli. So E. coli will adapt to its environment. So if O2 is present, so we'll say O2. So if O2 is present, which process will E. coli carry out? Which process gives E. coli the maximum ATP per glucose? Aerobic respiration, right? So when lots of oxygen is present, E. coli carries out aerobic respiration. How many ATP per glucose? 30, yeah. In bacteria, we're going to go ahead and say 30 ATP per glucose. Okay? Now, what's really cool about E. coli is that if oxygen is not present, you know, this is anaerobic environment, this is an aerobic environment, obviously. So E. coli has two choices for anaerobic metabolism. So if there's an alternate electron acceptor present, like nitrate, it will carry out anaerobic respiration. And you will recall anaerobic respiration, it doesn't make as many ATP. It makes less than 38 ATP per glucose, right? So it's not as much as aerobic respiration, but it's going to make a lot more than 2 ATP per glucose, which is the yield in fermentation, right? So if O2 isn't present, an alternate electron acceptor like nitrate is present, then E. coli will switch over to anaerobic respiration, make a decent amount of ATP. And then if there's no alternate electron acceptor, maybe, for example, nitrates aren't present, what can E. coli do? Carry out fermentation, right? So it'll grow slowly, right? And why is it growing slowly? How many ATP is it making? Just 2, right? So it's not going to grow very fast. But using fermentation, it can make 2 ATP per glucose. It can grow slowly using fermentation. Okay. So we just see that many of these bacteria, they're incredibly flexible. They're really adaptable. O2 present, let's do aerobic respiration. O2 not present, is there an alternate electron acceptor present? If so, we'll do anaerobic respiration. If it's anaerobic and there's no alternate electron acceptor, no problem, we'll switch to fermentation. Okay, so we can flexible they are. Really, really flexible. Okay, you guys. So believe it or not, that's a finish of chapter 5. Right? Okay. So what we're going to do then is we'll start the chapter 6 PowerPoint. But remember, you guys, we won't get through it. So what is your homework assignment? Your homework assignment is to do the worksheet, okay, that's on D2L. There's a key to the worksheet too, right? And hopefully the remaining PowerPoint slides would help for those that we don't get to. But my hope is, you guys, that we'll at least get down here. I'm hoping we'll at least get to molecular oxygen, why aerobic environments can be toxic, what kind of protective enzymes organisms have evolved. But I'm pretty sure we won't get to the effects of temperature, pH, or solute concentration on the cell. So these topics, read it in your textbook. Make sure you do the worksheet, okay, because it would still be fair to ask questions on these topics on the lecture exam, even though we're probably not going to get to it today. They're pretty straightforward. They aren't too complicated. All right, so just the overview of where we're heading. We're going to focus on bacterial growth. Right, so we're going to talk about how bacteria divide by binary fission. We'll talk about the standard bacterial growth curve. And then we'll talk about factors which influence growth, such as nutrients. And then probably, I'm hoping we'll be able to finish with the discussion of the influence of oxygen on the growth of microbes. Oh,